Good morning, everybody. So I think we are going to continue where we left off yesterday. And just to summarize, so far what I have said is that inflammation is playing a very important role. We talked about a lot of biomarkers of inflammation and especially NF-kappa B. And then we also talked about some of the agents that can control inflammation. So we are going to continue on this theme, that what are some of the other agents that can control inflammation. And the last one that we discussed was neem, Azadi Rakta indica. So neem is very, very important, and there are a number of things that have been identified from neem that can work for cancer and various other pro-inflammatory diseases. And here is another compound from neem called nimbolide, which also is working very effectively both in blocking NF-kappa B and in inhibiting the tumor growth. And where we implanted human tumors, in this case you can see colorectal cancer, human colorectal cancer, we implanted in mice and looked at the growth in mice and then gave it nimbolide compounds, and we could show that we could inhibit the growth of it. And it is working through the inhibition of pro-inflammatory microenvironment. And again, the paper is published, and it is published in Clinical Cancer Research, which is quite a very prestigious journal. And if any of you are interested to find out more about it, you can look it up. And if you need a PDF of the paper, that is also available to you. So now the next agent that I will like to deal with is something else. And this agent is kind of very close to my heart. Why? Because as you know that I went to a school or got my doctorate in University of California, Berkeley. And there is a building in the campus which is called Life Sciences Building. And in that building, there are some very, 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 very major discoveries that were made. Discoveries about cell culture, how to put cells in culture, was first discovered over there. The first that cell cycle, that is that how plants breathe versus how human breathe, that is called Melvin-Calvin cell cycle, that was discovered by Melvin-Calvin. The whole process of photosynthesis, what is photosynthesis, was discovered there. And nuclear bomb was discovered there first time. That was thrown on Nagasaki, Hiroshima. And that is also a place where they discovered a sex factor, and they did not know what to call it, and they called it vitamin E. Okay? Because if you don't take vitamin E, the person becomes impotent. Until that time, the only vitamin was known is vitamin D, and nobody knew anything after that. So they did not know what to call this sex factor, and they called it vitamin E. And that's how it came about. Okay? So this is the vitamin E called tocotrienols. So at that time, the structure was not known. And anything and everything that I am telling you it is described in this review. Tocotrienol, the vitamin E of the 21st century, its potential against cancer and other chronic diseases. So what I'm going to show you is there are two kinds of vitamin E. One is called tocopherol, and that has a 37,000 publications. Another one is called tocotrienol, that has only close to 1,000 publications. Okay, and what is the difference between tocotrienol and tocopherol? That here are 3 double bonds that are present in tocotrienol, but they are completely missing in tocopherol. So there is a lot known about tocopherol, and there is a very little known about tocotrienol. And they all exist in alpha, beta, gamma, and delta. There are 3 different forms, 4 different forms. Okay. So in that building, life sciences building, what was first discovered was tocopherol. Okay. And then there is a palm oil. There is a Malaysian Palm Oil Board, MPOB, and Malaysia is the biggest producer of palm oil. And so in that palm oil was discovered this tocotrienol. And I have been in their advisory board for a number of years. And just to show you where this tocotrienol comes from, so here is a palm, here is a rice, here is a barley, here is a oat, here is a wheat. All these are sources of tocotrienol. So you can see that. Okay. So in addition, you can see that here is a palm oil, 940 milligram per kilogram of palm oil, the amount of tocotrienol. And here is a barley, 910, and here is a rice bran, grapefruit oil, oat, hazelnut, maize, wheat germ. All these are the sources of tocotrienol. Again, just to give you an idea that where it is coming from. Okay, and so this is the tocotrienol, and what we found that tocotrienol but not tocopherol, and I mentioned you the difference between tocotrienol and tocopherol, that there are 3 double bonds that are present in tocopherol that are not that are present in tocotrienol but not present in tocopherol. And we found that it is the tocotrienol but not tocopherol that is the one that is blocking NF-kappa B and preventing preventing a bone loss and all the other things. Anything connected with NF-kappa B, only tocotrienol will work. Okay. Again, the paper, as you can see, is published in Journal of Biological Chemistry, and if anybody is interested, can look it up more in detail. So, if it is inhibiting NF-kappa B, is it working against cancer? And the answer is yes. So here, the role of gamma tocotrienol in the treatment of human pancreatic cancer. Among all the cancers known to man, pancreatic cancer is the most lethal cancer. And from the time of diagnosis to death is 3 months to 6 months. And if there is any cancer which is more lethal than pancreatic cancer, it is a gallbladder cancer. And that gallbladder cancer does not happen in America, that occurs only in India. And even within India, it happens only in North India. It does not happen in South India. And even in North India, it happens only in people who live around the river Ganges. So it's something got to do with the river Ganges. And that cancer is the most lethal cancer known to man, even more than a pancreatic cancer. Okay. And in India, there is only one doctor that I know. His name is Vijay Shukla, and he is the director of the cancer center in BHU, Banaras Hindu University. And he specializes in gallbladder cancer. And there's no treatment available. Okay? And there, the survival is in matter of days, from the time of diagnosis. But in the rest of the world, pancreatic cancer is the most lethal cancer. So that is the cancer we looked at, and we showed that gamma tocotrienol inhibits the pancreatic tumor and sensitizes them to gemcitabine. Gemcitabine is the standard treatment for pancreatic cancer. So if anybody gets diagnosed with the pancreatic cancer, what doctors will give them the chemotherapy which is called gemcitabine, and that is the standard treatment. So the tocotrienol will inhibit the tumor by itself, and then it will further sensitize them to gemcitabine by modulating the inflammatory microenvironment. And again, it says it all. And again, this paper is published, and it was published in another very prestigious journal called Cancer Research. And again, that this paper is available to you if any of you are interested to know more about it. So I'm kind of giving you kind of overview that that everybody should know about. So I think that that's another compound. So having said about several different compounds, and we have about over 100, and I mentioned you the list of all those 100. So I don't have the time to go through each one of them, but here I would like to switch this subject to spices. And there is an article that appeared in Cell, which is one of the most prestigious journal, and what it is about, the global demand for more affordable therapeutics and concerns about the side effects of commonly used drugs are refocusing interest on Eastern traditional medicine, particularly those of India and China. And spices is one of India is the motherland of all the spices. So therefore, this article deals with a lot of spices. And so we ourselves did a review that, again, if anybody is interested, can look it up. Chronic diseases, inflammation and spices, how are they linked? And this is again Ajay Kunnumakara, who is now a professor in IIT Guwahati, is the driver for this article. So it is published in Journal of Translational Medicine, and I highly recommend you to go over it. So whenever we talk about spices, this is what we are talking about. So you can imagine all this should look very, very familiar to you, that what I'm talking about. So as you know that I mentioned before, it was Christopher Columbus and Vasco da Gama. And these were the two guys in 15th century, they were looking for spices. And it was Vasco da Gama who came from Portugal. He found the right way and he ended up in India. Whereas the other guy, Christopher Columbus, who was from Spain, he went the other way and he discovered America. And he thought he was found India, but no, he found America. And that's why people who were living in America, they were called now American Indians. So you have Indian Indians and you have American Indians. So the guy who ended up in America, he found those Indians all right, but he did not find any spice. So there's no spice in America. Okay. So whereas Vasco da Gama, that who ended up in Calicut, and he is the one who found the spices, and that is way back in 1498. Okay. And as you can see that this is the route that he followed to reach Calicut, and here is Goa, and he was coming from Portugal. So these are again all the different kind of spices. So there are over 50 different spices. Okay, 50, not just one. And again, as I mentioned yesterday, that if you talk to people in America or even Mexico or a lot of other countries, whenever you talk about spices, they think it is hot. That's the only thing in their mind, that spices are hot. And by now, I'm sure all of you know that the only spice that is hot is red chili. The rest of them are not hot. So people get scared in the Western world whenever you talk about spices. So they talk about spices, but they never use spices. Okay? So we have done a book, and I got approached by a publisher, and this is a Barnes and Noble, which is one of the most prestigious publisher in America. So they published this our book called Healing Spices, How Spices Can Heal. Okay, and now this book has been translated into German, it has been translated into Italian, it has been translated into Spanish, all different languages, and it is available. You click on Amazon and you put it, and immediately you will get it. So, and it has been a very bestseller. Over 100,000 copies have been sold of this book. Okay. So we have another book, again, if anybody is interested, and the title is Molecular Targets and Therapeutic Uses of Spices. Again, this was done together with Ajay Kunumakara, and it talks about more science behind the spices. That what is there in spices that are active, and how does it do whatever it does. So again, it is all discussed in this book. So America uses a lot of terminology about spices, and the kind of terminology you hardly ever hear in India. So what kind of terminology? Terminology such as add spice to your life. Okay, and I hardly ever hear in India, but in America it is all the time, and it's very, very clear. And curry in hurry. British, as you know, were in India for a long, long time, and even British had a problem with spices because they could not remember the names. So what they did, they started mixing different spices together and gave them a common name, like garam masala. It has a 15, 16 different spices. Like curry, again, they combined several spices together and started calling it curry. Okay, even though the curry for the Indian people means something completely different, but they use the word curry. And so you will not hear curry here in India much, but you go to anywhere in England that they use it all the time. And so even terminology like curry in hurry, spice it up, spice queen, spice goddess, So this is all the kind of terminology, so much so that most of the Western women, for some reason, I don't know why, they are named after spices. Most of the women, which is kind of mind-boggling, And so, for example, that anise is a very common American woman name. Okay, ginger, very common American woman name. Okay, and rosemary, mace, pepper, basil, tulsi, sage, jasmine, angelica, curry, chili, tamarind. Yet They don't even know what is tamarind, but the woman's name is tamarind. It's very, very, very common. So much so that the woman who's trying to be the president of America, her name is Tulsi Gabbard. She is the governor from Hawaii, and her first name is Tulsi. Okay, and so we have an association called Hindu Economic Forum. Okay, and so what that is all about, it is an international organizations, and what it is all about, that what Hindus have contributed to the world economy. So there's a all world Hindu Economic Forum. So there was and they have their conferences every year in different countries, and about 2 years ago it was in Los Angeles, and Tulsi Gabbard was invited to give a lecture, and she gave a lecture, and before she started a lecture, she had a shloka from Bhagavad Gita. So she chanted that whole shloka before she started her lecture. So in other words, what I'm trying to tell you, that even she's a white woman standing on stage and talking about Bhagavad Gita, and her name is Tulsi. So that goes on to show you that how passionate some of these people are about the culture over here. And these are all the names that are adapted in America. And at least I do not know anybody in India, woman's named like this. I do not know of any woman in India that are called all of this. So we have another article, again, if you are interested, called Dietary Nutraceuticals as Backbone for Bone Health. So there we put all the spices together and all the various agents, that how they can work for you to fix your bones. Because as most of you, if not all, sitting here in the audience are very, very young. So you don't have any issue with the bones. But as we get older, bones loss becomes one of the major issue. So therefore you start having a backache, you start having a knee aches, and all different kind of aches. So bones started disappearing. So question is, what can you do to stop it? So that is where we came up with all the dietary nutraceuticals that can inhibit bone loss. And so basically that is dealing with all the spices. And Manoj Pandey, who is now professor in New Jersey, another product from my laboratory, so he has been the driver on it. So healing with spices, this is another student of mine, Julie Chung, and so here she put together all the different spices and what they look like, and you can see some of them, and asafoetida, this, that, and and it is there also in the other book, that healing spices. But I want to give you an idea, and most of you are here in the audience are familiar with it, but some of you may not be, and at least in America, hardly anybody familiar with it, and they may have heard of it, but they have no clue that what it looks like. And so I wanted to kind of share with you that what some of these spices are. So we have done a review now, another one, that targeting inflammation-induced obesity. And as I mentioned you, that no country is more obese than America. And so and even obesity can be targeted with spices. And metabolic diseases. And diabetes is one of the metabolic disease. And again, no country has more diabetes than America also. Okay, that how you can do that by curcumin and other different nutraceuticals. So here you can see this is what curcumin looks like, and here is a cinnamic acid coming from cinnamon. Here is a piperine coming from black pepper, and here is a capsaicin coming from red chili, and here is a isoeugenol coming from cloves, and here is gingerol coming from ginger. And so all these are different spices, and they are all targeting inflammation. And then these are all the molecular pathways for obesity, and that we have outlined. And here are the pathways that are linked with cancer and various other diseases. So we indicate that what are the various targets that are hit by it. So here again the same thesis that continues on, and that is a red chili, turmeric, ginger, black pepper, cardamom, cumin, and curcumin, capsaicin, all of them without exception. And we have shown that, and other people have followed up. They are all blocking NF-kappa B. Okay? And then when they block NF-kappa B, these are the major pro-inflammatory biomarkers, and they are all, as you can see, that they are inhibited. Okay? And then here is the IL-6 that is activating a STAT3 pathway that I already mentioned. And these are all the various biomarkers that have been linked with all these various chronic diseases, one by one. So again, by using spices, you can block all of them. Okay? So I think this is something very, very important I wanted to share with you, and I'm holding it off. And this is a paper just appeared, and it is amazing, I'm a little older than some of you, and I happen to be thinking about something that, okay, what have we done, what we should have done, what I have learned, and what I have not learned. So while I was thinking about that, there were some thoughts going through my mind that, did I do the right thing? And while I was thinking about all of that, all of a sudden, there is a paper that appears. And I had a very, very hard time believing that. And the paper that appears is, I get Indian Journal of Medical Research online. I get a lot of other journals also, but Indian Journal of Medical Research is another one that I get every week or every other week online. And this happened to be the issue, the latest issue of May 2019. And the title of this issue, of this article, is Need for Alternative to Animals in Experimentation and Indian Perspectives. And this is exactly the thought that was going through my mind. And what I was asking, okay, here is a cell culture. Here is a normal procedure is you go from cell culture, you to animals, and then to humans. And has there been any drug discovered based on cell culture? And the answer is no. Has there been any drug discovered based on animal model? And the answer is no. So then why are we doing it? And then, has there been any drug discovered based using human? And the answer is yes. But even there, what you are going to hear, the chances are 1 in 10. So which means if you test 10 drugs in human, only 1 will make it. So, but in cell culture and in animals, 99.9% failure rate. Okay, they simply don't work. So then why do we want to do it? Why we are wasting our whole life on doing cell culture and the animal when nothing is working? So having said that, so this is the article that deals with it, and it is by Soumya Swaminathan, Vijay Kumar and Rajni Kaul. It's the Indian Council of Medical Research, and I contacted this Rajni later on. And here are some of the facts. The facts are, and this is based on this article, that for drug discovery it is estimated that more than 100 million animals are used annually. 100 million. Okay? Animal data does not translate to successful clinical outcome. Then why are we doing it? Okay? There is a failure rate of over 90% between the nomination for phase 1 clinical trial and approval of new drugs. Phase 1 meaning where, okay, you have done the cell culture, you have done the animals, and now you go to human. And even when you go to human, which is called phase 1 clinical trial, which is called safety trial, that still 90% of the drugs fail even in a human. Okay? So, right now, fewer than 1 in 10 medicines that start being tested in human clinical trials succeed. So, you have to test 10 drugs in human for 1 to make it. Okay? So, poor translation along with increasing cost. So, what is increasing cost? $2.5 billion. $2.5 billion each drug. Okay? Or even more for drug discovery and development. Who has that kind of money? As I mentioned, that the total drug market in the world is $800 billion. And $400 billion is US alone. And India's share is only $4 billion. And here it costs one, drug, $2.5 billion to come up with it. India has never been able to come up with any drug. Okay, and here it is indicated within India 2 decades of drug discovery, and she considered only 2 decades of drug discovery R&D did not produce a single new chemical entity. Because the approach is wrong. And this is exactly what was going through my mind, and now I have some data to show it to you and share with you. I'm not talking hypothetically, and here are the people who have done their homework. Okay? So therefore, we all have to think, what are we doing with our lives and why? What's the point? Okay? And I think that is very, 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 very important. Okay? Simply because the kind of approach that is being taken. So, these are some of the drugs that come from Mother Nature, and you have aspirin, you have metformin, you have steroids, and this vincristine comes from Sadabahar. You have a statin, rapamycin, methotrexate, lithium, camptothecin, Taxol, artemisinin, doxorubicin, diclofenac, vancomycin. They are all derived from Mother Nature. Okay, and they are very, 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 very simple. And for example, that here is a plant called Sadabahar, and you can see what this plant looks like. And that is where it is called Vinca rosea in a botanical name, and that is where vincristine and vinblastine came from. And it is used even as of today. Okay. So, and then, as all of you know, Government of India created a separate department, and that is called Ayush, which is Ayurveda, Yoga, Unani, Siddhi, and homeopathy. And as I'm told, and I could be wrong, that they have come up with the first medicine for diabetes. First medicine. And what is that? It is called BGR34. And they have done even clinical trials here that I indicated for diabetes. So what does it have? It has a berberis ursitata, Tinaspora cordifolia, and here is another plant, Malabar quino. Here is the Gymnus sylvestra, which is called good mar. Good means sugar, mar means destroys. So traditionally it is called gur mar or sugar destroyer. And Rubia cordifolia, and here is Trigonella phonigicum, which is nothing but fenugreek. Fenugreek, methi. So they have put all of them together and they call it BGR34. And it is being used for diabetes. Okay, so something very, very simple and no side effects. Here is another one. You know, probably most of you who brush your teeth use Colgate, and Colgate has taken over. When I was growing up, I had never heard of Colgate. And we had only that neem. That neem stick is the only thing that we were using when I was growing up. And then with the time, Colgate has taken over. And now India is trying to make their own Colgate, because Colgate has become a giant, a mega. So we are promoting companies, you know, coming from other countries. That means we have no knowledge of our own. So here is a company called Vishnu Industrial Chemical Company. And that's what VICO stands for. Okay? And it was formed in 1952. Okay? I'm sure perhaps most of you may have heard of this VICO. Okay? And so this is VICO Vajradanti and herbal toothpaste. And you can see the number of plants they put together. And I have been totally hooked to it for last 5 years. My wife has been using it even for longer. So, you know, I mean, all kind of things in there, and that I think is as good as any. I have not been to a dentist in last 30 years. Otherwise, the normal American guy goes to a dentist every 6 months. So I have no issue. I still have all 32 teeth. So point of the matter is that try to be as close to the Mother Nature as possible. And try to promote your own products. And here is one of them called Vico Vajadanti. Okay. So I also mentioned you that the drug development cost, if you go the American way of developing drugs, here is AstraZeneca, here is the GlaxoSmith, here is the Sanofi, Roche, Pfizer, and you can see that probably you can see $11 billion for drug development, $8 billion by Glaxo for drug development. Here is a Sanofi, $7.9 billion. Here is a Roche, $7.8 billion per year for drug development. Now, who has that kind of money here in India to spend on that? So if you were to follow their route, you will never have a drug. So they are trying to recover their money in their own way. And that's not a win-win situation. Okay. So then I think most of you know what is a targeted therapy. Anybody who does not? So targeted therapy is that where you come up with a specific protein, a specific target you think is responsible for a disease, and then you find a chemical or an agent which hits that particular target, and if you can inhibit it, you think you have the cure. That is called targeted therapy. But the question that was asked, and this is the paper that came from as recent as September 11, 2019, just last week or 2 weeks ago. And this paper appeared in Science. Okay. So what they really found, that targeted therapy is not really targeted therapy at all. So there you could use a specific technology called CRISPR, where they can completely knock out the target and drug is still doing the same thing as it was before. You understand what I'm saying? So in other words, whatever the drug was doing was not through the target that it was designed for, it is working independently. Okay? So that is what it is. Targeted therapy truly targeted? And that's totally shocking. Okay? So 10 experimental cancer drugs killed cancer cells even when their intended target protein was missing. It is total nonsense. But this is the reality. Suggesting that drugs work by some other unknown mechanism. And virtually every disease known to man is multi-target, multi-genic. So just by hitting one target, you are not going to have a drug. So then why are you wasting all your time? Okay? So here is the evidence to indicate that some cancer drugs miss their target. CRISPR could improve their aim. That's the title of it. Okay? So I think this is very, very important. All of you, as I said, are very, very young. So you have to really think about all of that. Okay? So then how to discover drugs? And that's where I use the word serendipity. So what is serendipity? So serendipity is where you don't know what is happening and why it is happening, but it is happening. And that's why, I'm here in Nagaland, because I understand Nagaland has one of the highest number of tribes. And I'm told there are total 900 tribes in the whole world. And out of that, 700 are in India alone. And no country in the world has more tribes than India does. So much so that Delhi University set up a separate department of innovation. And what does that department do? So their approach is to go to the tribes and ask them that if you have this problem, what do you do? If you have that problem, what do you do? So try to collect the information from all the tribes and use that as a knowledge for drug discovery. Because they are already using it. And each tribe have their own concoctions. That's another way. And there you are going from humans, because they are using it in humans. So you are going from human to the test tube, to the laboratory, to find out how is it working. And that's what I call reverse pharmacology. And that's what I call serendipity. Okay? So we have listed a lot of things here in this review. Serendipity in cancer drugs discovery, rational or coincidence. So to begin with, it is a coincidence, but now you try to make a rational. How is it doing whatever it is doing? So you provide a rationale. So your job as a scientist, you can make a major breakthrough. Then you don't need billions of dollars to come up with a drug. You can have it overnight. And you will see a lot of drugs were discovered that way. So you just go ask your grandmother, hey, I have this problem, what should I do? Everybody will have their own concoction. And now you provide the scientific basis. So here is another one that I may have mentioned before. Cancer drug discovery by repurposing, teaching new tricks to old dogs. Okay, and I think I mentioned that yesterday. And so this is simply, taking the same drug for using A, B, C, D, whatever comes along. Okay, so you are finding the new uses, Like curcumin, I'm going to show you with curcumin, there are over 100 different uses. I have yet to find a disease where the biomarker is not altered by curcumin. And curcumin is just one of the examples. Okay. So, and then I think I also mentioned about the neem, Azadirachtica indica, and Indian traditional panacea with modern molecular basis. Okay. Again, we try to pick up with all the targets. And neem again has been around for thousands of years. So, and then there is a cannabis, and this is Cannabis sativa, and then there is a Cannabis indica, which came from India. In fact, that is one of the first one. And cannabis is very, 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 very common over here. And here they did the whole review, and these are some of the active compounds that are mentioned. And the paper just appeared in August 2019. And basically what they are finding, that this is the people from University of Gallup researchers, they found that there are pain relieving molecules in cannabis that are 30 times more potent than reducing inflammation than even aspirin. Just imagine, 30 times. How can you go wrong? So, and cannabis is so easy to grow. And all you have to do is start grinding up the cannabis and you start asking what is in it and how it is doing whatever it is doing. And there you have a product. Okay? And so these molecules are non-psychoactives and they target the inflammation at the source, making them ideal painkillers. So you simply cannot go wrong. You can cash in. So molecular targets of nutraceutical derived from dietary spices, potential role in suppression of inflammation and tumorigenesis. And again, very similar to thesis that what I'm telling you before. Okay, and this is also published. And to give you just few examples, that this is a turmeric, the active compound is curcumin. Here is a fennel, the active compound is called anethol. Here is a red chili, the active compound, here you can see the structure, is capsaicin. And here is a cloves, the active compound is eugenol. Here is a fenugreek, the active compound is diasgenin. And here is a holy basil, the active compound is ursolic acid. And they all block NF-kappa B. And all these spices should be very, very familiar to you. And simply we have shown that which spice does what and how. And this cannot hurt anybody. You don't have to give this to mice, to ask that does it work. You directly give it to people and find out about NF-kappa B. Anybody will take, fennel, anybody will take turmeric, anybody will take any of these things. So you directly go to people. Then you miss everything and you're directly getting the answer. Okay? So it's very, 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 very simple. And they all block NF-kappa B. Okay? So here, for example, red chili, and you can see what red chili looks like. And this is a capsaicin. This is what makes it hot, capsaicin. So it is a very potent inhibitor of NF-kappa B. Okay? And capsaicin, novel block, and it is also blocks STAT3 pathway. Remember I mentioned you NF-kappa B pathway and STAT3 pathway. And both the pathways are completely shut down. So very, very, very simple. No big deal. Okay? And here you can see this is, so the way we look at NF-kappa B, as I mentioned you, there are three different proteins, P50, P65 and I-kappa B. And I-kappa B undergoes phosphorylation and degradation. And then P50, P65 goes to the nucleus and binds the DNA. Remember that? So, and here we are looking at the DNA binding, and you can see as we add capsaicin that at 300 micromolar is completely no binding at all to the DNA. So this is binding comes down in a very dose dependent manner. Very simple. Okay, and here is a STAT3 pathway that is, remember I mentioned you that STAT3 undergoes phosphorylation and then it goes to the nucleus, binds the DNA, and here we are looking at the phosphorylation, and as you can see as we add red chili, capsaicin, this phosphorylation goes down and down and down, and at 50 micromolar there is no phosphorylation at all. So very easy. So you can do this kind of testing, you know, and very, very easy to do, you know, and that is, this is purely intellectual, but bottom line is you want to see what happens in humans. And anybody will take chili, and then you just look at the urine and saliva and look at all these biomarkers. So that is the ultimate goal of it, which I highly recommend. So, and here is a ginger, you know, and again, ginger is very, very harmless, and a lot of people love ginger. And the active compound in ginger that here we tested, it's called zarambone, you know, and this is what the structure of that zarambone looks like, and this is what ginger looks like. And we showed that zarambone abolishes NF-kappa B, and this is the kinase, I-kappa B alpha kinase. Remember I mentioned you that this is the kinase that causes the phosphorylation. Okay, and so it is completely inhibited by ginger, activating, leading to suppression of anti-apoptotic and metastatic gene expression and upregulation of apoptosis and downregulation of invasion. All the right thing. Just we provided a proof with ginger. And again, published in Oncogene, and you can look it up, there is no problem at all. So it is this kind of thing, and then once we have shown that it inhibits NF-kappa B, next step is what happens with the bone loss. And here you can see this is NF-kappa B is completely inhibited by ginger, and here we are looking at the bone loss. This is a control, and this is with zarambone. So we can prevent the bone loss by using ginger. So anybody who has a problem with their bones, just tell them eat more ginger. That's it. So there your cost is not an issue, safety is not an issue. All you are saying that just eat a little bit more ginger. It will not cost you millions and billions of dollars. It is that simple. Okay. So again, the paper here, as you can see, that we published it in Cancer Research way back then, and you can look it up. And here is a black cumin, kala jeera, you know, and this is what that looks like, and this is the active compound from black cumin, and here is a suppression of NF-kappa B altogether, and here we can see the tumor in the control, that how the tumor keeps growing. And here, moment we give a black cumin, and you can see that the inhibition of the growth of the tumor after giving black cumin. So it is again very clear. And we published the paper targeting NF-kappa B activation by thymoquinone. So this is the compound called thymoquinone in suppression of anti-apoptotic gene products and enhancement of apoptosis. So here is another one that we published. Thymoquinone inhibits angiogenesis and tumor growth through suppression of AKT and extracellular signal regulatory kinase signaling pathway. So it is this kind of the proof of the concept that you can provide. There is absolutely no problem. So how much time we have more left? I'm sorry? Okay. So I think that we can continue. I don't want to kind of bore you too much, and so I have a lot more data, and so we can continue, and we can stop right here and ask more questions. So more questions you ask, the better. I just want to make sure that all of you understood what I'm talking about. And then if you understood, you will be able to do something for your future. And that's what it is all about. So please ask questions. So we stop right here. I'm sorry?
How is the ginger helping the bones?
How what?
You said that ginger helps for bone health.
I'm sorry?
Ginger is helping bone health.
Yeah.
So at what level is it? What cells?
So the question is, ginger is helping bone loss. And question is how? Am I right?
What level? What level?
So question is at what level? So there are osteoclast and there are osteoblast. So osteoblast are required for bone formation and osteoclast are required for bone loss. So the osteoclastogenesis, the bone loss, is completely inhibited by ginger. Okay, and that is working through the activation of a NF-kappa B pathway, which is inhibited by ginger. Okay. So that is what we mean by osteoclastogenesis, meaning the osteoclast which are required for the bone loss is inhibited. Yeah.
What's good for the liver cancer?
Anything and everything.
Yeah, I mean, is there something to do with the biomarker? Because liver is the first of our organ which meets our nutrition nutrient diet. So does it have any connection to that in your journey to reach?
Yeah. See, keep in mind that no matter which cancer you talk about, and I showed the data earlier that NF-kappa B is playing a role. I have yet to find a cancer where NF-kappa B does not play a role. Okay, so liver cancer is no exception. So if you inhibit NF-kappa B, you will be helping liver cancer. And the question is how to inhibit NF-kappa B. Again, I covered a lot of agents about it. So far I have not found a cancer which is not linked to NF-kappa B. Okay, without exception. So you name it. So you go to a doctor, doctor is not a scientist. So therefore, they go through all different routes. Same thing, you go to pharmaceutical companies, they are just trying to sell the drugs. So they will go through all different routes. So point of the matter is their approach is not going to help you at all. Their approach is trying to how to make money out of you. You are giving them the opportunity to make money. Okay. So any other question? Please feel free. The more question you ask, the better.
Sir, can we use these drugs during pregnancy?
I'm sorry?
Can we use these drugs during pregnancy?
Can you what?
During pregnancy.
Can we use these drugs during pregnancy?
Can we use these drugs during pregnancy?
The answer is yes and no. Okay. So during pregnancy, one of the compound is HCG, human chorionic gonadotropin, and that is one of the hormone which is produced, and that is also, by the way, blocks NF-kappa B. We showed that. And during pregnancy, I cannot think of any reason why not. Meaning, I cannot think of any reason how it could be harmful. I'm just telling you offhand my personal opinion. But has there been any study? I'm not aware of it.
Actually, sir, in some literature like dioxin, actually obtained from the diasporia plant, it is generally used as like a modification drugs by the some tribes that will do dioxin.
Diabetes, yeah, tinospora? You are talking about tinospora? No.
Diosgenin.
Yeah, diasgenin, that is fenugreek.
It's been used by some tribals.
Some tribes
Yeah,
from the modification.
there you are. So that will block NF-kappa B, diasgenin. You are talking about diasgenin. And that is coming from fenugreek, methre. That is a methre. Diasgenin is coming from methre.
Actually, mainly they recommend like you have talked regarding let be your food, your medicine, and medicine be your food. Actually, in just one place. So in regard with that, so during pregnancy, actually, is there any rules or times when we market this type of food like spices as drugs? So we just give that this is suitable for the during the pregnancy or adolescent days like that. Is there any rules? Is there any approved rules for like?
So what is the question?
You mentioned the word diet and
Diasgenin?
He said food is medicine. So in pregnancy, what type of food we should recommend?
are there
Is
any
there some
rules?
Are there what?
Which all diets are convenient during pregnancy at all?
Yeah. So, to be honest with you, offhand, I cannot give you any answer. I have to look it up. If you have a specific question, feel free to write me, bbaggarwal@gmail.com. I can look it up, but I personally do not see any of these agents can potentially harm during pregnancy. I cannot think of any.
Sometimes actually in our home, my mother generally says don't keep such type of spices in the iron pot or in the brass. So is there any scientific evidence of that one? You don't have to keep in the particular metallic pot like that.
These kind of spices in his hometown, his mother says that these spices should not be kept in metal vessels.
Spicy should not what?
Kept in metal vessels.
Metal vessel. Oh, okay.
Is there any link, scientific evidence regarding that, not to be kept?
The answer is again yes and no. You know, I'm sure that there are metals that can mess up the system, and that's why people do what they do. But the question is there any scientific evidence? And the answer is not that I'm aware of. Yeah.
So the spices that you have mentioned that will be properly. Taking it raw and cook does it change its effective or activity doesn't change. Does the spices change its effectivity or activity when taken raw or cooked?
I think somebody has to help me translation.
The spices that you have mentioned that limit NF-kappa B, or the spices that you have mentioned, taking it raw and cooking, does it make any changes?
जो spices हम NF-kappa को inhibit करती हैं, should we take them raw or should we also use in cooking, which is the better?
So, is there a difference
Okay,
in raw
okay.
form and cooking
Yeah,
form?
okay. Thank you. So, I understood. So, there is again no evidence to indicate that by cooking you are destroying its potency. If anything, it is the opposite. And it has been shown, at least in case of curcumin, there are papers have been published that cooked turmeric is more active than uncooked. Okay, so whether that is true for all the spices, I cannot say, but there is no evidence to indicate it hurts. So the answer is yes, you can cook with it with no harm done.
So I just have a simple question. If suppose in case of diogenin, the same the structure is shown there. So I think it's audible. Okay. If the same structure, if we chemically synthesize in a chemical lab, is there any like a reversible or a non-reversible reaction caused in our body?
In principle, the answer is no. It should be same. There should not be any difference. And again, the only spice that we know the most about, and that is turmeric, haldi, and the active compound being curcumin. So that has been synthesized, and several different analogs have been made. And in fact, there is a company in Hyderabad that sells a synthetic curcumin in kilogram quantities, And so there is absolutely no problem whatsoever, and it is as good as any. Yeah. Any more questions? Thank you.